and welcome to Science Wrap, the show where we feed you the tastiest new research from the world of science, all wrapped up in an easy-to-digest package. My name's Miriam Guskin, PhD and science lover. Unless you've been hiding under a soil aggregate, you'll know that the Nobel Prizes have been awarded this week. In the field of chemistry, the prize went to these three gentlemen: Thomas Lindahl, Paul Modrich, and Aziz Sankhya. They've mapped at a cellular level how DNA gets repaired. Now, as you probably know, DNA codes all the information needed to create a human being. The way it does this is through a massively long series of organic molecules known as nucleotides. These pair up with their corresponding partner to form the two strands we know as DNA. Although it's tightly packed into our cells, DNA is actually massively long and could stretch to the sun and back 250 times. This sheer length, along with everyday copying or external factors like UV rays, mean that DNA is constantly being damaged and needs to be fixed. Figuring out how this repair works takes time and people. Thomas Lindahl's piece to the puzzle was flying in the face of the thinking of the 1960s and proposing that this DNA damage actually occurs. In 1974, he published a paper which identified a particular enzyme, N-glycosinase, which cuts out the damaged sections of DNA. Piece by piece, or nucleotide by nucleotide, Lindahl formed an image of how this cutting process, which he called base excision repair, occurs. This was the perfect starting point for the next big player, Aziz Sankhya. Through many years of work, he was able to isolate and characterize three particular enzymes that would cut and repair DNA in the way that Lindahl described. He aptly named these UVRA, B, and C. Three enzymes that worked in a team to coordinate this process. But what was Paul Modrich's part in all of this? He also worked with enzymes, in particular the DAM methylase enzyme found in viruses. This enzyme clung to particular nucleotides on the DNA where a miscoupling of the bases had occurred, and it acts like a little signpost to show which pieces of the DNA needed to be cut out. He characterized this whole process, dubbed mismatch repair, in viruses before moving on to humans. So three extraordinary scientists, three key processes: base excision repair, UV damage repair, and mismatch repair. Okay, but why is all of this important? Understanding the fundamental processes of DNA helps us understand genetically inherited diseases, cancer, and even aging. The other big Nobel news of the week is all about neutrinos. Neutrinos are basically the ghosts of the physics world. No, really, they're also known as poltergeists. They are tiny subatomic particles with no electric charge that can pass through ordinary matter undetected, and until recently were thought to have no mass. There are three kinds of neutrinos, each of which relates to another subatomic particle that does have an electric charge: electrons, muons, and tau. These little guys come from the formation of our sun during the Big Bang, but are also being made on a daily basis by exploding supernovas or even in nuclear power plants. Since about the 1960s, scientists have had a pretty good estimate of how many neutrinos were made by our sun. But the measurement of neutrinos here on Earth is far less than this estimate. So where did all the neutrinos go? Enter Takaaki Kajita and Arthur McDonald. The hunt begins in a giant neutrino hunting bunker 1,000 meters below Kajita's lab. It's basically a giant swimming pool filled with the cleanest water you can imagine. Neutrinos can pass through the tank and every now and again collide with a water molecule to form their significant other: an electron from an electron neutrino, a muon from a muon neutrino. On the other side of the world, below McDonald's lab, a similar bunker was carrying out similar experiments. This particular observatory could measure the neutrinos given off by the sun today, which are only electron neutrinos. They could also measure the total number of neutrinos at any one time. What was weird was that the number of electron neutrinos was far less than the number of total neutrinos in the tank, when really they should be the same. What this told McDonald is that these sneaky little guys were changing their form on the way from the sun to the Earth. And what this meant is that neutrinos had to have mass. Okay, again though, why should we care? Because neutrinos are formed by stars and the workings of the universe, they help us to understand the beginning of time, and also where our universe is heading in the future. So that's a wrap on some of the science news from this week. Feel free to subscribe to stay up to date.